So the sample size for a clinical study is really going to depend on the stage of evidence development that you're at. I'm going to focus on a pivotal study where you really want to make sure you have a sufficient sample size to draw a reliable conclusion at the end of the study. And there are 4 ingredients in a sample size calculation. The first one is the natural variability in the endpoint that you're studying. Whether that be a blood pressure reduction and just how variable that is, the standard deviation of that measurement, or the proportion of patients that respond to your intervention, or perhaps the rate of occurrence of heart failure hospitalizations in a heart failure population. That natural variability is background noise that needs to be overcome with a big enough sample size. The second ingredient is the effect size. How big of an impact does your device have on that endpoint? And then the third and fourth ingredients have to do with your tolerance for 2 kinds of statistical errors. The first one is often referred to as alpha or type 1 error. That's the chance of the data turning out to be statistically significant even when there's really no effect of the device, and that has to be held very low. Lastly, the power of your study, your probability of reaching a statistically significant conclusion at the end, that's that fourth ingredient that controls type 2 error.